Okay, could somebody confirm that you can hear me?
Can hear you.
Great. Okay, are there any questions? That wasn't what I meant to do. All right, if there's no questions, let's begin. I'll share the screen. Oh, that's taking time. So hopefully you can see the screen. We're going to be hopefully finishing chapter 2 today and get started with a little bit of chapter 1. Please note that, is that right? No, I'm on the wrong week. I'm sorry, we're supposed to be working on chapter 2 today. Today's Thursday, August the 14th. And we just started chapter 2. I doubt we'll finish it today. And today we're going to have a lab 3 in the lab. Any questions about what we're doing? If no questions, let's begin. I don't mark where we were last time, so let me see if I can remember. I remember we had talked about water.
Yeah, we had just stopped talking about water.
So I talked about this slide here.
Yep.
Okay, great. All right. Let's talk about a few of the properties of water because water is an important molecule for life. And as I once stated already, the majority of life actually is, many living organisms are mainly composed of water. Water is not an organic molecule. It is an inorganic molecule, but organic living things are mainly composed of water. First property we want to talk about water is a good solvent. All polar molecules and ions dissolve in water. For a polar molecule, you can have something like sugar. As you know, you put sugar in our tea or a coffee. Coffee and tea are mostly water. And you can just put sugar in water and it'll dissolve in water. It's because sugar is a polar molecule. Ions also dissolve in water. An example is you put salt in water. The salt will dissolve and become sodium chloride. I'm going to talk about that. And dissolve in the water. By dissolving in the water, this would be a crystal of salt, and the big yellow circles are the chloride ions. The little green circles are the sodium ion. Sodium has a positive charge, chloride has a negative charge, and the salt crystals stick together in a crystal of salt. And if you could put that in water, the water will surround the ions. Oops, I go to the wrong one, sorry. I'm trying to blow this up so it'll be easier to see. Scroll this over. And water is a polar molecule, and the negative charge of the oxygen will orient, of the water, will orient itself towards the positive sodium ions. And that is why ionic molecules dissolve in water, because water will surround the ion with the negative, the opposite charge of the water, and this is a partial charge here, will orient itself towards the opposite charge here, and this is a full positive charge. And so in a sense, this shouldn't be drawn like this because this is a partial charge, that's a full charge. And that looks like a full negative charge. It's not, it's a partial negative charge. And I mentioned that your textbook author, I think it calls it a partial negative charge. Let's see, what's the other word that you use? I don't remember. And then for the negative ion of the chloride, the opposite end of the molecule will face the negative charge, meaning the hydrogen, positive hydrogens will orient so that they're surrounding the negative chloride ion. And that is why ions dissolve in water. The same thing happens for polar molecules, except like when you have a sugar crystal and you put it in water, the sugar will have partial positive and partial negative regions of the molecule. And then the water will orient to that partial charge, like the partial negative charge in sugar, the positive areas of the water will orient around the partial negative charge of the sugar. And then the partial positive charge of the areas of the sugar will have the negative region of water orient around that positive region of the sugar. Any questions about any of that? Anyways, the important thing to remember is that water is a good solvent. It can dissolve many different molecules if they are polar and if they are non-polar. Now, obviously, non-polar molecules like oil do not dissolve in water. So it's a good solvent, but it doesn't dissolve everything. Non-polar molecules will not dissolve in water. Polar molecules are hydrophilic. That means they're water loving. That means they have partial positive charges and partial negative charges, or if they're ionic, they have a full positive charge and a full negative charge. And all of these molecules are hydrophilic. Non-polar molecules are repelled by water, and we call them hydrophobic. Literally, phobic means fear of water. They do not dissolve in it. And oil is a hydrophobic substance. When you shake vinegar and oil together, they will dissolve and the oil will get on top and the vinegar will get on the bottom. Any questions about any of that? Hydrogen bonding between water molecules makes water excellent at buffering the temperature. All molecules absorb heat when you raise the temperature. And when that molecule gets hotter in its temperature, how you measure the temperature is just, the temperature is a measure of the molecule's movement. And so when you heat up a molecule, it will move around more. And that could be just moving back and forth or bumping around or whatnot, or its energy of motion. But water can absorb heat from both the molecules moving more, and then water can also absorb heat, and heat is an energy, and that energy can go into the hydrogen bonds. And the energy will break those hydrogen bonds, but it takes energy to break those hydrogen bonds. And so water can absorb heat in two ways. You can break the hydrogen bonds, and you can make the water molecules move around more. Okay? And then when water molecules evaporate, the water molecules with the most energy are the water molecules that are evaporating. So that keeps the body of water cool, because the water molecule with the most energy is the molecule that is evaporating. And that is why water is excellent at keeping us cool when we perspire, because the water molecule that is evaporating is the water molecule with the most energy, meaning it's taking the most heat away from us. Okay? And then, of course, it also takes energy to break the hydrogen bonds in the molecules. And so with all of that, water is excellent at buffering the temperature. And most of you guys probably know this, because in Western Washington and Western Oregon, our temperature is buffered by the water from the Pacific Ocean. And that is why in Western Oregon and Western Washington, our temperature doesn't vary as much as it does in Eastern Washington and Eastern Oregon, because we're next to the Pacific Ocean, and that water buffers the temperature. And so when a cold front moves in, our temperature stays warmer, milder than it does in the eastern parts of Washington and Oregon. And then in the summer, they get much warmer because water buffers our temperature. Any questions about that? Okay. Water can split apart. And when water, which is H2O, splits apart, it breaks into H plus ion and OH negative. H plus ion is a proton, and then OH minus ion is the hydroxide ion. And these will be charged particles in water. Because water does break apart, that is why, or part of the reason why, water can conduct electricity. Why, if you're on like a golf course and it's during an electrical storm with lightning, you should get off the golf course, assuming it's raining, because if a lightning hits the golf course and you're anywhere near that, you'll get a charge from the water carrying the electricity from the electric bulb. And if you don't know, salt water is even worse for carrying a charge. And that's why you should get out of salt water like the ocean if ever there's a lightning storm. The reason why salt water carries more electricity, more charge, is besides water breaking apart, in salt water, we have sodium ions and chloride ions, and they can carry a charge. The charged molecules in water can participate in chemical reactions. And when water breaks apart, that gives water the pH. And we'll talk a little bit about that. pH is actually the measure of the hydrogen ions in concentration in the water. So that's what pH means. Any questions about that? All right, I don't see all of this. Let me scroll that up, I think. Nope, too far. pH is measuring the hydrogen ions in solution. The more hydrogen ions you have in solution, the more positive charges you have in the solution. The positive charge comes from the H plus of, well, water or something breaking apart. And with more H pluses in the water, we call that acidic. On the other hand, if there's more OH minus hydroxide ions in solution, then the water will have more of a negative charge, and we'll call that a basic solution. You can also call that an alkaline solution. Now, when you have distilled water, before CO2 gets in it, and it first breaks apart, water will be neutral. And that's because when one water molecule breaks apart, let me show you that. It'll break apart into one H plus ion and one OH minus ion. That is neutral because the H plus concentration equals the OH minus concentration, or you can say the protons equal the hydroxide ion concentration. And that is a neutral pH. What that is pH 7. Any questions about that? Now, in normal situations, when we have pure water, it doesn't remain pH 7 for very long. Does anybody know why? I'm not hearing anything, so I'm going to assume nobody wants to admit that they know why or they don't know why. Water will change its pH over time and become around pH 5. And what's happening is CO2 from the air gets dissolved in the water. Let me show you. Oh, I didn't go into it here. So we have H2. I'm just going to do that above. Oh, oops, no. Will absorb CO2. There's my arrow. And these molecules will come together and become H2CO3, which will split apart, giving off an H plus ion, and I'll try and make that an ion, plus HCO3, oops, no O there, O3 negative. And I'm going to superscript that to make it a little clearer. And superscript this right here. Any question about all of that? So this is what will happen when CO2 dissolves in water. And as you can see, we will get this CO2 releasing some hydrogen ions from the water. And then the other molecule that's made is bicarbonate right here, which is a negative charge. This is actually important in your blood because this is a major Oh, how do I end that? Maybe that ended it. This is a major buffer in your blood. And this can go into H. I guess I'll just go, why don't I just copy that? Well, I'll do that. I'll do it this way. This is the correct way to do it. 2 H pluses. And then plus, come on. Don't know if that would work. There we go. Get rid of that H. Now, this would happen under fairly basic solutions, this breaking apart. And so in our blood, we mainly have this and this. But if we needed more hydrogen ion in our blood, this could break apart and form this. But this is just one of the buffers in our blood. And the purpose of this buffer is to maintain the pH of our blood. And for example, when people drink a lot of soda or something else that's very acidic, that will increase hydrogen ions in solution. Let me see how I can get that over here. So we're going to add hydrogen. That should be a plus. Which I know a lot of you drink soda, so this would be an example. So we're getting hydrogen from our diet or our drink. And what that's going to do is, I don't know if I can do this. Well, maybe I can do it right here. So it'll make this go this way. That didn't work well. And then this reaction will go this way. And then this reaction will go this way. And that should be all together in a line. I don't know why that's lower than that, but we'll just go with it. And what you're doing is when you eat or drink something that's acidic, your buffer will move this way and then move this way. And you see what happens when this forms that? It'll form CO2, which then you will breathe out. And the other thing you form is water, which just becomes a part of your blood. And this is just a buffer in our blood to help the blood keep its pH, the correct pH. Any question about any of that? All right. Anyways, here's the pH scale. It ranges from a pH of 0 to a pH of 14. I mentioned that pH 7 is when the H plus concentration in water equals the OH minus concentration in water. And I will state a little bit more, and that is that pH equal to 7 actually means that one water molecule, which is H2O, will break apart into H plus and OH minus for every one. I'm hoping you can see this because I'm doing it at the bottom of the slide. For every 1 times 10 to the 7th, I don't know if I can superscript that there. That's color, don't want that. Anyways, that's 10 to the 7th power. And I don't see how I can superscript this, so we'll just leave it there. Maybe that's it there. Any questions about that? So that 7 actually means that there's 1 hydrogen ion for 10 to the 7th water molecules. And a pH 8 means there's 1 hydrogen ion for 10 to the 8th water molecules. And then pH 14, which is extremely basic, you would never come across that. Although oven cleaner is approaching it, it's about a pH 13.5. pH 14 means you have 1 hydrogen ion for 10 to the 14th water molecules. And then a pH 0 means you have a 1 hydrogen ion for 1 times 10 to the 0 water molecules. And that is 1. So at a pH 0, the water molecule breaks apart and we have an H plus ion. Any question about any of that? All right. So an acid is defined as a substance that dissociates, giving up 1 or more hydrogen ions or an H plus ion. An example of an acid is HCl. When you put HCl in water, the HCl will break apart into H plus and Cl minus. And that is why HCl is an acid, because it gives off hydrogen ions. Now your textbook defines a base as a substance that dissociates and gives up an OH ion, the hydroxide ion. An example of that would be sodium hydroxide is a base because when it dissolves in water, it will dissolve as sodium ion and hydroxide ion. So this will be given up and that'll make the water basic. But I do want you to know that your textbook either oversimplified or made a mistake, because a base is a substance that either gives up OH minus or it absorbs H plus. An example of a base that absorbs H plus is bicarbonate. Like you put sodium bicarbonate in water, it will absorb the H pluses and then make the solution more basic as the H plus joins the bicarbonate to become, I think this word is bicarbonate right there. Any question about any of that? All right. So your textbook is a little wrong on what a base is. A salt is a substance that dissociates in solution, such as water, but when it dissociates into a cation, that is an ion with a positive charge, and an anion, an ion with a negative charge, neither of which are H plus or OH minus ions. So sodium chloride is a salt, and it'll dissociate into sodium ion and chloride ion. And I think you guys all knew that sodium chloride was a salt, but so are magnesium chloride, for example. It is a salt, but we don't call it that because in the generic common usage of salt, we only use it for table salt, which is sodium chloride. But in science, the word salt is any substance that dissociates into a positive cation and a negative anion. And here we see sodium chloride as a solid dissolving in water as sodium and chloride ions. Any salt is an electrolyte. An electrolyte is a substance that when it dissolves in water, it can conduct electricity. And so all salts are electrolytes. Any question about any of that? All right. And that's why salt water is better at conducting electricity than pure water, because it has the sodium and the chloride ions. Okay, organisms need to maintain a fairly constant balance of their cellular acids and bases to remain healthy. And as I stated, the pH is a measure of the H+ ions in solution. It's actually exactly the measurement of pH is equal to the negative log of the hydrogen ion concentration. I'm not going to ask you about that, but pH is equal to the negative log of the hydrogen ion concentration. And I've already said the pH scale is between 0 and 14. Between 0 and 7, we call it an acid, not 7, between 0 and 7. And then between 7 and 14, we call it a base or an alkaline. And most organisms grow best between a pH, well, around pH 7. So between a pH of 6 and 8. And your textbook says exactly 6.5 to 8.5, but I think it'd be better to say around pH 7. Most organisms, not all organisms. All right, any questions about acid and bases and salts, anything like that? If there's no questions, I'm going to move on. Talking about organic compounds. We have several organic compounds shown here. You don't have to memorize any of them, but I do want to state that organic compounds are usually held together by carbon bonds, like all of, let me blow that up a little so you can see it better. Too far. Boy, I moved that way too far. Didn't know I moved to another slide. There we go. So all of these bonds are hooked together between carbon, with the exception of these two right here. And carbon is the main molecule hooking together organic compounds. Organic compounds always contain carbon, did I say that right? And they always contain hydrogen. They frequently contain oxygen, but they do not have to contain oxygen. Like there's oxygen there and there and there and here and here. But here we have an organic compound, which is a simple organic compound. And we call these simple organic compounds hydrocarbons. Hydrocarbons only contain carbon and hydrogen. And these are organic compounds, but they're also hydrocarbons, which are a simple organic compound. You can draw out the organic compounds this way, or you can use the circular structures. And if you don't know, wherever there's a point, that means there's a carbon there. If ever there's a molecule that's not a carbon, you have to write it in. These are hydrocarbons, so all of these are carbon. And wherever there's a double bond, that means this carbon and this carbon are connected by 2 molecular bonds. So these carbons are connected together with 1 single carbon bond. These carbons right here have 2 carbon bonds between them. Any question about any of that? If there's no questions, let's talk about organic compounds. Like I said, they always contain carbon and hydrogen. They may, or many of them do contain oxygen, but they don't have to contain oxygen. They may contain other elements like nitrogen, sulfur, phosphorus. All of them are held together in an organic compound by covalent bonds. Organic compounds can have other bonds, but the main bond is the covalent chemical bond, but they may especially have hydrogen bonds. Organic compounds are typically more complex than inorganic molecules, and they're capable of more complicated biological functions. What are some examples? Well, an organic compound has functional groups attached to it. So methane, which has a hydrogen here, when we put an OH group on it, that gives this molecule different properties than methane. And this we call a functional group because the OH group gives this molecule different properties than it would have without the OH group on it. For example, methane is a gas and it doesn't dissolve in water. Methanol is a liquid and it does dissolve in water. And you probably know that because ethanol is drinking alcohol and it looks pretty much like water. And it certainly dissolves in water. Carbon is in the main molecule in organic compounds, and carbon can bind to 4 other atoms. Like this carbon binds to a hydrogen here, a hydrogen here, a hydrogen here, and then it binds the 4th place to an OH group here. They can be bound, the carbons, like this, or like this, and you can make them longer like that, or they can be bound in the ring structure, which I've already shown you here. There's a ring structure.
That's a benzene ring.
Say again?
That's benzene, right? Benzene structure.
This one here is close to, let me read it. I got the names down here. This is close to cholesterol, but it's not because something's missing from it. That is close to benzene. It's not benzene.
Okay.
I think benzene is one
ring.
One ring, yes.
Okay.
So this is benzopyrene, if I'm reading that correct. All right, so let's look at some examples. Already talked about that. I need to talk about, because carbon is holding the molecule together, we say that organic molecules generally have a carbon backbone, because carbon is what's holding the molecule together. We already talked about the functional groups. Shrink that. Functional groups are what are responsible for most of the chemical and physical properties of particular organic compounds or organic molecules. I mentioned OH being a hydroxyl group in alcohol gives the alcohol its properties, such as its hydrophilic properties, why it will dissolve in water. So OH attracts water molecules to it and helps alcohol dissolve in water. Here are some other functional groups you do need to be familiar with the groups shown in a red box. We already mentioned OH is alcohol. NH2 is an amino group, and it's found in proteins. This is the rest of the organic molecule attached to the amino group. And R just stands for essentially anything. Okay, any question about any of that? You should know the carboxyl group. It is C bound to oxygen with a double bond, and then bound to a hydroxide group with a single bond. So you can state that as COOH. And that's the carboxyl group. It is found in proteins, lipids, and organic acids. And then the last functional group you should know is phosphate. It is PO4, meaning 4 oxygens. And it orients like this, where the phosphate has, in this case, 2 negative charges. You can find phosphate groups in ATP, DNA, and nucleotides. Why do you think I want you to be familiar with the groups in the red boxes? Nobody has a guess?
Is that because we are learning about lipids, DNA, proteins?
Yeah, it's because these groups are the groups found in the biological molecules. Okay, so these are important groups that you can find in the biological molecules. Of course, OH is found in a lot of molecules, but NH2, mainly in amino acids and proteins. Carboxyl group, mainly in proteins and lipids and organic acids, which are close to lipids. And then really phosphate, mainly in nucleotides, DNA, and RNA. Oftentimes, the molecules will have more than one functional group in the molecule. For example, in an amino acid, we have both the amino group, NH2, and carboxyl group on the amino acid. Any question about any of that? All right, when we're talking about organic compounds, it's important to realize that the small organic molecules can combine together to form large macromolecules. The macromolecules we call polymers. Poly means many, poly, mer, I'm not sure what mer means, many units. So many units. And the units that make up the polymer, we call those small repeating molecules monomers. So monomers come together to form the polymers. And many of the biological molecules are polymers, like starch, the polymer of sugar, and proteins, a polymer of amino acids, DNA, a polymer of nucleotides. So all of them, polymers, are made up of the monomers, which would be nucleotides for DNA, amino acids for proteins, and various sugar groups for carbohydrates, such as starch. How that happens is in a dehydration synthesis reaction or a condensation reaction, taking 2 monomers, coming together, and then they're linked together to form the polymer. And this linkage is called dehydration synthesis. Synthesis, you're making a bigger molecule, or condensation reaction, you're condensing these molecules to form water, where condensation comes from. Anyways, that's joining the monomers. You'll note that when your monomers, the ROH, comes together with the HR, and the R can stand for essentially anything organic in this case. They come together so that the R binds directly to the R, and the H comes off of this monomer, and the OH comes off of this monomer in dehydration synthesis to form water. Okay, and then the reverse reaction can happen. We can take the polymer, like starch, add water to it, and then we can undergo hydrolysis to form ROH plus HR, meaning where we form the monomers. And the polymer has at least one monomer come off. Let's make that the monomer. And then this right here is actually a smaller polymer if we're going this way. And actually you should be familiar with hydrolysis because Marty McFly said to his mother, "Ma, you sure know how to hydrate a pizza." And what she did was she took a hockey puck-like substance, put it into her hydrolysis machine of the future, hydrolyzed it, adding water to it, and then out came this pizza. Remember that? Nobody's commenting. Most students actually like that tie-in to Marty McFly. Dehydration is the mechanism for synthesis of macromolecules, such as how we make carbohydrates, lipids, and proteins. And let's take a look at that in a little different form. Dehydration synthesis, making a larger molecule. This is actually a polymer already, but we're adding a monomer to it. And the water comes out, OH on this molecule, H on that molecule. The monomer then binds directly to the polymer and making a larger polymer, and that forms water. And then the reverse, you add the water to the polymer, and this monomer breaks off, grabbing the OH of water, and the H of water joins to the smaller polymer right here. So hydrolysis and condensation synthesis reaction are the reverse of each other. And in our body, we do this all the time. Like if you eat a high carbohydrate diet, you will add glucose to your glycolysis. Is that the right word? I shouldn't have given this example because I'm not positive of what that's called now. I'm getting a brain malfunction. So let's go ahead and say a green plant will add on sugar to a starch molecule, and then they'll get a larger starch molecule. And that would be a condensation reaction. And that's how green plants like corn will undergo photosynthesis to make sugar, and then take that sugar and then make starch out of it. And that's where we get cornstarch from. Any question about any of that? Now this one I can say happens in your liver, and so does this, but I forgot what this molecule is called right now. And it might be glycolysis, but I'm not positive of that. I'll have to, I think I have a picture of it. Glycogen? No, it's called glycogen. Anyways, this one happens in our liver. And whenever we need sugar, because you need sugar in your blood, like you're exercising a lot, and your liver will break the polymer, adding water, and then a monomer will come off, and that will add sugar to your blood. Any question about that? and this molecule being glycogen in your sugar or your muscles. All right, carbohydrates is the first of the biological organic molecules we want to talk about. These include, of course, sugar, starch, and the other complex carbohydrates like cellulose. Carbohydrates are an important energy source. Cells and organisms use them for structure, and you can find carbohydrates in nucleic acid. Carbohydrates consist of carbon, hydrogen, and oxygen, but they have a general formula, CH2O, and then you put a parentheses around that and N, meaning that for every carbon in a carbohydrate, we have 2 hydrogens and 1 oxygen. So like for glucose, it's C6H12O6. Any question about any of that? So carbohydrates include simple sugars or strings of sugars linked together. A starch is a very long string of sugars hooked together. Lactose or disaccharides are 2 sugars hooked together. And we can classify carbohydrates into 3 major groups based on the size of the molecule. You can have monosaccharides. They're the smallest carbohydrates, and they're a single sugar group. And then there's disaccharides. That's 2 sugar groups hooked together. And polysaccharides are long, big molecules composed of many sugar groups. Any question about any of that? Monosaccharides are simple sugars. They all range in size from 3 carbon atoms to 7 carbon atoms, but most monosaccharides are either 5 or 6 carbon atoms, like ribose is a 5 carbon sugar, and glucose and fructose are 6 carbon sugars. Monosaccharides, typically sweet, and they're water soluble. Disaccharides are formed by joining 2 monosaccharides together in dehydration synthesis. Although the disaccharide can be broken into monosaccharides by hydrolysis. So here we have glucose and fructose. They're both monosaccharides. Let me blow that up a little. And we pull water off of glucose and fructose, and then we directly link this carbon to this oxygen right here. And then we have the disaccharide fructose and glucose is, it's not lactose. Oh, that's sucrose. It's right there, just needed to look. Sucrose, okay? And then we can eat sucrose. Sucrose is table sugar, and we'll digest that by adding water, breaking this bond here in a hydrolysis reaction to form fructose and glucose, the 2 monosaccharides. Any question about any of that? All right, the polysaccharides consist of many monosaccharides. They can be straight, a straight chain like starch, or they can be a branch chain like glycogen, which is similar to starch, just the chain branches. Some examples of polysaccharide include starch, glycogen, cellulose. They're all polymers of glucose. meaning starch, glycogen, and cellulose, are all made up of the simple sugar glucose. And they are covalently bonded differently, but other than that, they're the same. Chitin, I'm not really going to talk about much, but here we're looking at some starchy foods: bread, cereals, pasta, potatoes, rice. I think you all knew that. Beans do contain starch. Beans are about a third starch, same with most nuts, about a third starch. But beans and nuts also contain protein. So they're about a third protein. And then the other third is both beans and nuts contain fat. That's why soybeans, you can get soybean oil out of it from the fat in the bean. You don't need to know any of this picture, but it's showing you the different polysaccharides. Let me briefly show it to you bigger. Get that out of the way. I'll move it down here. A starch, mostly it's a non-branching chain like that. It can occasionally, very rarely does it branch. Glycogen, the same, except that this is a branching chain. You can see a branch here, a branch there, a branch there. Cellulose, which isn't very visible here. Let me see if I can blow this up. I can't blow it up more. Is a link of glucose hooked together here, and then another chain of glucose hooked together here, and a chain of glucose hooked together here. And it's not, I can't show this. Let's end the slide, see if I can blow it up better this way. This yellow line, that's not a covalent link. This is a hydrogen bond between this glucose and that glucose. And if you notice, in cellulose, there are a lot of hydrogen bonds linking the chains of glucose in the cellulose together. And that's why cellulose is so difficult to digest and why most animals cannot digest cellulose.
I'm sorry. I thought humans cannot digest cellulose, but animals can. Because we have
Only certain animals can. Animals that are ungulates, that have more than one stomach, or rabbits, which they actually eat their food twice, meaning they eat the grass and it passes through their digestive system. And the first time it passes through, they eat the first turd so they can digest it a second time. Okay.
Thank you.
Those animals can digest cellulose, but in reality, they are not digesting the cellulose. They have microbes in their stomach, which are digesting the cellulose. And I think I've mentioned this before. A cow cannot digest grass, the cellulose, directly. The cow has a bunch of microbes in their stomach, and it's the microbes which are digesting the cellulose. And they are digesting the cellulose and then making some of that sugar available to the cow. And that is symbiosis. The cow benefits because the microbes are breaking down the grass, and the microbes living in the cow's stomach benefit because they get a home, the stomach, and they also get fed grass when the cow eats. And so they share some of their sugar with the cow, because if they don't, the cow would starve and then they'd lose their home. And that's just symbiosis, the two living together. Any question about any of that? I won't test you at all on symbiosis, at least for this point right here. All right, let's talk about the second of the biological molecules. And the second of the biological molecules are lipids. They consist of carbon, hydrogen, and oxygen. And although they do consist of oxygen, you should realize that there's very little oxygen in a lipid. Let me show you the next slide. I'll come back to this slide. Load this up. So this is a side chain of a lipid. And as you can see, there are many carbons and hydrogens, but there aren't very many oxygens because this OH actually comes off when this binds to make the lipid. And so all there is for here is one oxygen. All the rest are carbons and hydrogens. The point is, there is oxygen in lipids, but there isn't very much oxygen in lipids. Lipids are, with one exception, non-polar molecules and insoluble in water, meaning they are hydrophobic molecules, with one exception. Lipids are the structural and energy storage molecules for cells and for organisms. Lipids are a primary component of the cell membrane, and we'll talk a little bit about that when we talk about the molecule that makes up the cell membrane. We can break lipids into three groups: simple lipids, complex lipids, and steroids. Any question about any of that? If not, let's talk about simple lipids. Simple lipids are made up of fatty acids, a major component of lipids. A fatty acid consists of a fairly long chain of carbons and hydrogen. It does have one carboxyl group at the very end, but most other locations, hydrogen is bound to the carbon. We blow this up. A saturated fatty acid is one where the carbon only has single covalent links between the carbon. So right here, this is a saturated fatty acid because there's only single carbon links. In palmitic acid right here. An unsaturated fatty acid is a molecule having at least one double carbon bond between the carbons. Let me show that here. So this is a molecule of fat, which is a lipid. And as you can see, this is an unsaturated fatty acid or an unsaturated fat because there's one double carbon bond right here in the fat molecule. All the other bonds are single carbon, but there's only one, and we call it an unsaturated fat because it has one double carbon bond. You can notice that wherever there's a double carbon bond, there is a kink in the molecule, meaning this molecule isn't flat like that one. It comes to here and then it bends. I think that's what we had to talk about here. The unsaturated fatty acid bond causes a kink in the chain. All right. The most common simple lipids in your body are called fats or triglycerides. They're called triglycerides because they have 3 fatty acid chains right here, right here, and right here attached to glycerol. So there's the fatty acid. Let me blow that up a little. There's the fatty acid chain. It links to glycerol. And in dehydration synthesis, we have water coming out, and the OH comes off the carboxyl group, and a hydrogen comes off the glycerol. And then that will link directly this oxygen of glycerol to the first carbon of the fatty acid chain. Any question about any of that?
So is that a ester, that oxygen, is that a ester group?
An extra group. I'm not sure what you mean by an extra group, but this
No.
oxygen is part of glycerol. And when you link glycerol to a fatty acid, you have the oxygen forming these 2 links right here. So does that answer your question?
I meant the functional group, ester functional group.
No, ester is the type of linkage. I'm not going to talk too much about that, but when we have eukaryotes and bacteria making fatty acids, no, that would be true for an archaea also, but we'll talk about archaea. They form an ester linkage between the O and the carbon. And this is just a chemist term for this link. So whenever you have the COC, they call that an ester link. And that came about from the hydrogen leaving the glycerol here, and then the OH leaving the fatty acid here, and then this carbon binding directly to that oxygen. So I think I mentioned triglycerides are named because there's 3 fatty acids and 1 glycerol. Oh, and we should say that this is an example of a monomer, 2 monomers, a fatty acid and glycerol coming together to form a triglyceride. Triglycerides are the body's richest energy source. It's stored as adipose in animals. And if you wanted to see some triglycerides, just look at your belly. I guarantee there's triglycerides in your belly, no matter how thin you are. And if you're not very thin, you will have a lot of triglycerides. A slight mention about the body's richest energy source. If you needed a lot of energy all at once, or let's say you're in Point Barrow, Alaska. I think that's as far as north as you can get in Alaska, whatever is far north of Alaska. And then your car dies and you got to go hiking through the wilderness to get to town in order to survive in the cold weather. And so you want to carry with you something to eat to give yourself energy. The best source for getting something to eat to give you more energy per weight of the molecule is to eat a lipid. You'll get more energy from that than, well, let's say 1 pound of lipid than you would from 1 pound of carbohydrates or 1 pound of protein. Now, I'm not saying that eating lipids is the healthiest thing to do, but if you need the energy, you should eat lipids. And actually, if you eat the correct lipids, it is healthy. And we'll talk a little bit about that. All right, any questions about lipids? Some lipids include oils, which look like that or like this. And that's why they're liquid, the oils. Examples of other lipids include butter, solid cream, lard, and they will be composed of molecules like this. One lipid we need to talk about is the phospholipid. It is the main molecule in a cell membrane, actually in any membrane, but especially cell membranes. Let me blow this up a little bit. You'll note that a phospholipid has two fatty acids. And we call these tails to the phospholipid, the blue part, which is the fatty acids. And we're not going to really talk about it much, but all phospholipids, at least human phospholipids, have one saturated fatty acid and one unsaturated fatty acid. And I shouldn't say all phospholipids are that way. I should say phospholipids of humans are that way. Because if you have a microorganism growing in some place that's really cold, the microorganism will have more unsaturated fatty acids. And then if you have an organism living like in the tropics, you'll have fewer unsaturated fatty acids. Any question about any of that? But we're not talking about humans living in the tropics or humans living in the poles. I'm talking about animals. All right, no questions about that. Let's go on. The phospholipid, the fatty acid tail, is bound to the fatty acid head, which is right here. And the fatty acid head composed of glycerol right here. Let me blow that up so you see it better. Glycerol right here. But instead of like a triglyceride, you don't have 3 fatty acids, you have 2. And then the third position of glycerol doesn't bind to a fatty acid. It binds to a phosphate group. And then there will be an organic molecule, organic group attached to that phosphate group, attached to the glycerol. This region of the fatty acid, excuse me, the phospholipid, we call the head region. This region of the lipid, we call the tail region. And remember, this is a particular liquid, the phospholipid. Why this is particular lipid is that it is an exception from other lipids. All other lipids, are hydrophobic, meaning they do not dissolve in water. The phospholipid can dissolve in water. Phospholipids are a molecule where this region here is polar right there and ionic right here. And so this region will dissolve in water. This region is nonpolar and it will not dissolve in water, but it can dissolve in lipids such as oil. So this molecule can dissolve in both oil or lipids, like cholesterol or fat, this molecule could dissolve in it, and those would be solid lipids. but it can also dissolve in water because of the phosphate group. So this makes this molecule biphasic. It can dissolve in both water and oil. And there aren't too many molecules which are biphasic. One that you're probably familiar with are detergents. It is a biphasic molecule. And one region can dissolve in the water. Other regions can dissolve in the lipid. And that is why detergents help clean our clothes, because the water can dissolve out the polar molecules and the ionic molecules in our clothes, but water cannot remove the oils from our skin or any other oily or fatty material in our clothes. But the detergent can, because this region of the molecule will dissolve in the oil or fat, and this region of the molecule can dissolve in the water. All right, any question about any of that? Well, this is the picture of the molecule showing you the exact chemical makeup. Let's shrink it down to here, showing us the phospholipid, and it has two tails shown there, and then a head shown here. When we put phospholipids in water, this region will not want to be around the water. This region will be wanting to be around the water. And what the molecule will do is it'll just start lining up, where this molecule will be lined up with that molecule, lined up with that molecule. And then on the other side of this, I don't know, linking or chain of phospholipids, we'll have another row of phospholipids with the fatty acid tail on the inside, and then the phosphate head of the phospholipid on the outside. And then that will just keep lining up until we get them lining up like this. And that'll keep lining up until we get that or that. This I want to talk about a little bit because this should look familiar to you. What does this look like?
Cell membrane.
I didn't hear that.
Cell membrane.
Yeah, this looks like a cell, with this being the cell membrane. And in the cell membrane, the main molecule is actually the phospholipid, and it'll just line up one after the other, with the heads being outside the cell, because if we're like thinking about blood, outside the cell is mostly water, and that's actually true of any of your body. Most of the molecules are water. And then the phospholipids will be on the inside, because inside the cell is cytoplasm, which is mostly water, and the heads like to face the water. The tails want to stay away from the water, so they come together, because lipids can be with other lipids, but they don't like being around a polar molecule such as water. And that's why we get phospholipids. If you put a whole bunch of molecules in water, you'd eventually see them taking on this shape. Before that shape, they take on this shape. And before that, they would take on, I don't have it shown here, but they start forming this. There's something else I need to talk about there. All right, because animals only have the cell membrane, they don't have a cell wall, animals tend to put cholesterol in their cell wall. And the purpose for that is that the cholesterol aids in strengthening the cell membrane. And so you can find cholesterol in the cell membrane of animal cells. You do not find cholesterol in the cell membrane of plant cells because a plant cell has a cell wall. It doesn't need to strengthen the cell membrane. All right, any question about any of that? If not, I'm going to stop here and we will continue with this next time. Any questions? All right, I'll see you in the lab at 6:30.